The slide, you are not given slide, Smith sir.
Sir,
You will give slide later, right?
sir, your voice is not audible, sir.
The slide, you have to give slide now. Later on you will give, like after sharing the video, right? Smith sir, am I audible? Smith sir. Hello?
You are audible, sir. You can start. You are audible.
Okay, fine. Thank you, sir. Thank you.
So good morning, one and all. Is my voice audible, sir? Address, sir.
Yes, sir. Yes.
So welcome all the participants for the 1 week online FDP program at Sri Vishnu College of Pharmacy. Today, Dr. Kabimani sir is with us for the national FDP. So with the topic of the tiny fish with big impact. So with this, I would like to I would like to ask Dr. Principal sir K.S. Nataraj to address the gathering.
First you play the college video, Sunil. Sunil, play college video.
Yes, sir.
Can I start?
Sir,
Okay, thank you, Dr. Sunil. Respected today's speaker, Mr. Kavimani sir, and my fellow teachers and my dear students, and my warm welcome to 1 week online FTP SBP program entitled Emerging Technologies in the Field of Healthcare and Pharmaceuticals on 4 to 8 December 2023. On behalf of students and staff and management of the Vishnu College of Pharmacy, Bhimavaram. The main objective of the FDP is to bring awareness on present technologies in healthcare and pharmaceutical field. My sincere thanks to all speakers for acceptance of our invitation in spite of their busy schedule. I wish you all the best to the faculty and students. Make use of all the sessions, and you can find many takeaway points from each and every session. Okay. Once again, I'm very thankful to Professor Kavimani sir, who accepted our invitation, even though in spite of his busy schedule at the university. Okay. Thank you, sir. Thank you. All the best. And over to Sunil sir.
thank you, sir. With this, I would like to invite the day coordinator, Dr. A. Srinivas Rao sir, to give the introduction about the speaker.
Good morning to today's participants and the speaker, sir, Dr. S. Kavimani garu, and my dear colleagues and students. Very good morning. I would like to introduce Dr. S. Kavimani garu for this welcome to the seminar. He is currently working as a professor and head of the Department of Pharmacology at the Mother Teresa Postgraduate and Research Institute of Health Sciences, Pondicherry. Sir graduated from MMC College Madras and did his PG at Bombay from Jodhpur University, Calcutta. He is a Gates Scholarship holder and he has 30 years of teaching and research experience. He guided about 44 PhDs and currently guiding 6 PhD students. He has total 265 publications to his credit. He written 2 books and 5 chapters in Elsewhere and Springer publication books. He is a member of Expert Committee for Pharmacy Council of India, AACP, New Delhi, and MBA. He is a member of the Board of Academic Committee in Pondicherry University, MS University, Jawaharlal Nehru Technological University, Kakinada, and Annamalai University, Krishna University. He is also a visiting faculty for Pondicherry University and MS University. Central Council member of, he is a Central Council member of PCI New Delhi, Chairman of the BPharm Syllabus Committee, PCI. He was an approved CPS CEA nominee for various institutes. He received Best Teacher Award from Antiviral Society and received Best Pharmacist Award from the MGR University. Received Best Teacher Award from IPGA. Thank you for coming to the today's part of FDP, sir. Even though you have the hectic schedules, thanks for accepting our college invitation, sir. Thank you, sir. The session is over to the coordinator of the FDP program.
Thank you, sir. Am I audible?
Yes, sir.
Yes, sir. My slides are visible.
No, sir.
No, sir. Please start sharing.
It is not visible.
Not visible, sir.
Okay, 1 minute. Is it visible?
Yes, sir.
Okay, good.
Visible.
Yes, a very good morning to one and all. I'm very happy to be a part of Sri Vishnu College of Pharmacy, which organizes the 1 week online FDP program on emerging technologies in the field of healthcare and pharmaceuticals. And my sincere thanks to the Principal K.S. Nataraj sir, Coordinator Bobby Raju sir, and Organizing Secretary Y. Randi sir for giving me this wonderful opportunity. This small introduction, I would like to start my lecture. And the theme of the title of my lecture is the tiny fish with a big impact. So why I have chosen this particular topic? Well, just I want to give an introduction. There is stringent getting permission from the CPCSA and the Institutional Animal Ethical Committee for doing research on rodent like mice and rat. And the cost which we are spending for purchasing of rat, per rat it is 360 rupees. And for transportation, we have to spend much more money. So instead of going for rodent, some research or some experiments that can be carried out by using the Zebrafish. So the 5R concept in alternative to animal experimentation is reduction, refinement, replacement, then rehabilitation and responsibility. In this 5R concept, I will be discussing about the alternative to not animal, it is alternative to the rodent. So I have chosen this particular topic, the tiny fish with big impact. So now if you see here, so for doing research, for the seventh sem we have practice school, and eighth sem we have mini project work. And for the batch of 100 students, what project we have to carry out? Suppose if 100 students are divided into 25 batches, then what kind of project work we have to carry out, either in the practice school or in the project work? So this kind of lectures, either by in vitro models or by in silico model, or even by in vivo model by using the Zebrafish, and that will be useful for the teaching faculty. So in that concept, in case of in vitro models, the cell cultures can be used. Number of cell cultures are available for in vitro studies, MTT-assay or DPPH-assay, something like that. There are some advantages of using cell culture. It is easy to work with, and the maintenance cost is very low, and it have a short experimental cycle. The only thing is it has to be maintained properly in laboratories. And there are some experiments that can be carried out in fruit fly and mus plus, as you know, conventionally we are doing project work with mus plus. But my interest is on zebrafish. Zebrafish have high reproductive rate, and all the organs will be developed in externally. So embryo development which takes place in externally, and it is so transparent, so that we can go for embryogenic toxicity studies, etc. So I will be explaining more details in forthcoming slides. Now, I would like to give introduction about the zebrafish. It belongs to family Cyprinidae. The genus is Danio and the species is Rario. Danio rario. The advantage of this fish is, it can be alive even in ordinary water as well as in saline water. In seawater, it can be in seawater also and in ordinary water also. Even though many number of fishes are available for experimentation study, here we can say in zebrafish itself, 40,322 papers have been published in PubMed index journals. That indicates that zebrafish is widely used fish model for the research purpose, either for therapeutic uses or for toxicity studies. If you see this histogram, comparative statement from 1997 and 2022, when in 2022, this I have taken in the last month, that means in the month of October. So October itself, many number of papers have been published by using the zebrafish model. This is with respect to the PubMed. And now I would like to discuss about the similarity between, say, human and zebrafish. So why I want to say is, in 1960s, there was thalidomide tragedy. Administration of thalidomide in pregnant human crossed the placental barrier and interfered with the embryogenesis, organ development. That is called phocomelia. But in rat or mice, the thalidomide fails to produce teratogenic activity. That means thalidomide is toxic, teratogenic in human, but not in animals. Arsenic, it is highly poisonous to human, but it is not at all poisonous to rat. Morphine is CNS depressant in human, but it is CNS stimulant in cat. Penicillin, even though produces anaphylactic reaction in human, but in cat and guinea pig, it produces death. Penicillin is a life-saving drug. Penicillin is a life-saving drug in human, but it takes the lives of the animals. So there are so many dissimilarity between the rodent and human. But here I would like to give a similarity between the zebrafish and the human. The 70% of the genes which is present in the human is also present in the zebrafish. Moreover, 80% of the gene associated with the pathophysiology of the diseases in human is also associated with the zebrafish. And we can say here, liver, pancreas, gallbladder, circulatory system, digestive system, lungs, central nervous system have similarity between the human and zebrafish. Now, I would like to give some of the characteristics of the zebrafish. This adult is 3 to 4 centimeter length. It is sexually dimorphic. So how will you differentiate a sex? I will be giving in the next slide. The lifespan of the zebrafish is 3.5 years, and the sexual maturity is 6 to 8 months. Optimum breeding can take place between 6 months and 18 months. So these are some of the characteristics of the zebrafish that will be useful for our studies. For example, if you see, one mating between the fish can produce approximately 300 eggs in each conjugation in one night. mice and rat and all, the fetuses or the litters will be 6, maximum 10, not more than that. But in zebrafish, it is above 300. And these embryos can reach the larva stage after 3 days of post-fertilization. Within 3 days, the eggs can be converted into larva. Within 3 months, the larva will be converted into an adult. So, the reproductive cycle is very quick. The small eggs can be placed into the wells of the plate and survive for approximately 7 days without any feeding also. No need to change the water, no need to give any feeding, but eggs can survive approximately 7 days in a petri dish. Now I would like to give how will you differentiate a male with a female. If any pharmacologists are there, I think this lecture will be useful for almost all the staff members of pharmacy. So the distance between the anus and the reproductive organ, for example, vagina. The distance between vagina and anus is less than it is said to be female. In case of rat and mice, I am saying. The distance between the anus and the reproductive organ is more than it is called male. So this is the way how to differentiate a male rat with a female rat. Whereas in case of zebrafish, you can say this is male. I think you can able to see my cursor. Male have a slim golden body with the dark blue stripes on it. Whereas female, you can say a curved region. This is the curved region. No, this is absent in case of male. The female have a large round abdomen. This is called a large round abdomen. Even if you purchase fish for picking purpose for your home, we can differentiate which is male fish and which is female fish. A silver undertone on lighter blue strips. Here it is dark blue stripes. Here it is light blue stripes. This is a female and this is male. And if you see here, the urinary tract, which is given in yellow color, testes and ovaries. This is in case of male, testes and ovaries, and this is in case of female, testes and vas different. This is ovaries and oviduct in case of the female. Suppose if you want to see toxicity studies, embryogenic toxicity or reproductive toxicity, then we have to choose only the female fish. That's why I'm giving how to differentiate the sex. Now, the fish can be classified according to its size of the fin. This is called the short fin, and this is called wild type, and this is called long fin. The number of fins you can see here is a fin. So why I want to say this? There is one experiment, growth promoting activity, like growth hormone, like activity can be studied by using this fin that I will explain in later slides. Now you see here, there are 5 different types of fins are there: pectoral fin, pelvic fin, anal fin, dorsal fin, and caudal fin. And these 3 are single and these 2 are in pair. So suppose if you want to study the growth promoting activity, the caudal fin that can be cut. You can cut here, right? So now it is finless, caudal finless, or the size of the fin is less. After administration of the drug, which is said to be growth promoter, you will find the growth of fin. Okay, that also I will explain in later slides. Now, how to choose the fish for our experimentation purpose, how it should be. This is called body condition scoring for adult zebrafish. You can see here BCS 1, 2, 3, 4, and 5. In BCS 3, the body larger than head. Body should be larger than head. That is said to be fish is well conditioned. Whereas head and body is equal in size, that is called under conditioned. Head larger than body. See here, see BCS1, head larger than body, it is called emaciated. When head and body is equal, then it is called under conditioned. When body is larger than head, that is called the fish is well conditioned. So we will be choosing the well conditioned fish for our study purpose. Now, if you see what is the life stages of the zebrafish, you can see I have given the length of the fish as well as the maturation in days. Fertilization, after within 3 hours, the embryo will be converted into larvae. This is embryo hatching, and it's converted into larvae within 3 days. The larva will be 3.5 millimeter in length. Within 24 days, the larva may be converted into the juvenile. That is called metamorphosis. The juvenile can be converted into adult, that is called sexual maturity, within 120 days, within 4 months. So it is better to use 4 months old zebrafish for our experimentation purpose. Now, see comparative statement of embryonic development in zebrafish, rat and human. For example, I will be taking only one, craniofacial development. Craniofacial development, which takes place on 8th day in case of rat, whereas 22 days in case of human. In case of zebrafish, this is on 22nd hour. Able to understand? Craniofacial development takes place within a day in zebrafish, whereas 8 days required for rat, 22 days required for human. So early embryonic development in zebrafish, so that organogenesis studies, embryonic toxicity studies, teratogenic potential, reproductive toxicity studies. So everything can be carried out in zebrafish in a short span of interval. Now, I'm going to discuss the embryonic development. You can see here, this is within 20 hours of fertilization. You can see this video, there is development of embryo. I think no problem in viewing the video. So this is the time-lapse video, shows zebrafish development from a single cell to 22 hours after fertilization. So what is there in green color? I will explain afterwards what is green color, etc., etc. So now you will find a complete the embryonic development in this video. Now you can see blood flow in zebrafish, trunk, head, and heart of 2.5 days old zebrafish, and you will find the flow of fluid inside the fish, and this is the zebrafish heart. Blood flow in red color, and blood vessels are shown in green, blood cells are in magenta, and here you can see the beat of the heart. So blood vessels are shown in green color, and blood cells are shown in magenta color. I think this will give clear idea about the zebrafish. Now, how to identify whether it is a healthy conditions or not? In fish, whenever you are getting animals from the breeder, quarantine for 14 days, then it has to be checked by a veterinarian for presence of zoonotic diseases, etc. And by observing the rat or mice, you can say whether it is sick or not. Then how will you find out whether the fish is sick or not? The help of an appearance or with the help of behavior. And this is probably due to the poor water quality and housing and management. For example, in case of appearance, fins held close to the body, changes in fin structure, slugged mucus, changes in color pattern spot, weight loss. Like that, you can give so many examples. Whereas in behavior, you see, the fish will try to jump from the tank or swimming near the water surface. There will be loss of balance, not eating, slow swimming, changes in shoaling behavior, increased aggression, rubbing body on tank vessels, swimming near the bottom of the tank, or sudden death. So these are the behavioral abnormalities when the fish is having some infections due to bacteria, parasite, and virus. I said the tiny fish with big impact. So what you are going to do with this tiny fish? Many number of compounds can be screened to find out the target, either by using genome-based assays or disease pathology assessment. That is called molecular screening. With respect to acute, subacute, subchronic, and chronic toxicity studies, the toxicity assessment like nanoparticle toxicity, chemical toxicity, environmental toxicity, drug toxicity, and metabolite toxicity can be studied by using the zebrafish physiologically based pharmacokinetic model, genomic studies, toxicokinetic studies, and drugs for the treatment of neurodegenerative disorders, drugs which produce a statogenic effect, etc., can also be studied by using this tiny fish. The advantages of this fish is already I said high fertility and transplant embryos, rapid development and vertebrate, inexpensive, and genetic engineering as well as high throughput screening methods are possible by using this zebrafish. Now you can see here applications of zebrafish model in toxicological evaluation. Many number of, it have regeneration capacity of heart rate, blood vessels from form, how angiogenesis taken place. Is there any cardiac abnormalities, morphological abnormalities in liver, color changes, atrophy degradation, hypothalamic pituitary thyroid axis? Cancer can be studied in embryo, heart rate and circulation, brain regions to form cell movement, and in germ cells, gonad index, in nerve cells, biochemical index, neuronal necrosis, and kidney injury, kidney failure, and renal malformation. So all these toxicological studies are possible by using this zebrafish model. To give an example, I have given two embryos here. The first embryo, A, is the normal embryo. We will find well-defined eyes, brain, ear, and muscles. Whereas this is the embryo which is exposed with methyl mercury chloride. Methyl mercury chloride is highly toxic to the embryo. It is embryo toxic. So this methyl mercury chloride on embryo of 4 to 24 hours post-fertilization, you will find head and trunk structures are severely malformed. So if anybody is having some synthetic molecules, synthetic chemicals, then synthetic chemicals can be tested against the embryo toxicity. Now I would like to give 10 facts of zebrafish. You can say abstract of advantages of zebrafish. Almost all of the fish used in medical research are zebrafish. Zebrafish have the unique ability to repair large portions of damaged heart muscle. The regeneration capacity is having this particular fish. If the heart is damaged, it can able to regenerate it. Most fish can see in color and use color to camouflage themselves, attract by means of color. For the first 3 months of their lives, zebrafish fry have transparent bodies, allowing researchers to examine them internally without invasive measure. No need to go for invasive experiment, just by observation. Because within first 3 months, the body is very transparent. So if it is transparent, you can visualize the externally without doing invasive things. 84% of the genes known to be associated with human disease are found in zebrafish. Genetically mediated disorders like cystic fibrosis, cancer, or diabetes mellitus can be studied conveniently by using this zebrafish. There are several varieties of zebrafish. Zebrafish embryos are nearly transparent. I said research in zebrafish shows that fish can feel pain and even rub the area that hurts, just like we do. It have that capacity. So these are all some of the advantages of zebrafish. In 2003, the genome is sequenced in zebrafish. Now see, there are some zebrafish labs in India, well-established zebrafish labs. Institute of Genomics and Integrative Biology, Center for Cellular and Molecular Biology, Indian Institute of Science, Institute of Life Sciences in Hyderabad, National Center for Radioastrophysics in Pune. These are well-established zebrafish labs. Even they will reply if you send the mail for any help or any doubt. They will reply. So now the power of zebrafish is to model disease and therapy. I have said, here you see, this is the fin. The fin is you can cut the fin like this. So upon administration of the drugs, fish have the regeneration capacity. This is in larvae. This is behavioral screening. This is embryo development, chemical screening, and also cancer genocrypt model. I will be explaining 2 or 3 experiments to you. Now, if any pharmacology staffs are there, they can they can follow my procedures to practically demonstrate the effect of local anesthetics, general anesthetics, anti-anxiety drugs on zebrafish. And these videos I have taken in my lab itself. And this is anesthesia in zebrafish, the side view, etc. I have given everything I have given now. So now I want to show you some of the videos. So now first we have to anesthetize the zebrafish. Anesthetize the zebrafish for removal of blood sample or for removal of the brain. So how will you anesthetize the zebrafish? Just this is the cold water, this is ice water. So now you can see in ice water, we are putting the fish. So immediately it will get anesthetized. Immediately after anesthesia, the fish has to be removed and transferred into the normal water so that it will survive. So now the fish is added in the ice water, right? It is ice water. Now you can see the anesthetized zebrafish. Now almost it is anesthetized. Now we have to see. Now we have to change it in the normal water so that I can continue our experiment. Now it is anesthetized. So now it has to be transferred into a normal water. Okay. Now, local anesthetics by using the lignocine. You can see the fish will turn upside down. We have to see the carefully. This is lignocine You can see one rotation. Again, I will be playing the video. Now it is anesthetized. Last slide with ice water. This slide is with lichnocaine. Pharmacology lectures, please listen. 2% lichnocaine solution. You take 2 ml in 100 ml of water. Then put the zebrafish inside it. Again, you have to see the video how the animals turns its upside down. Upside down. Now you can see upside down. This is the rotation, right? If this rotation comes, then you can say the local velocity. Now there is lack of swimming. Lack of swimming, right? Now it is anesthetized. So this you can keep as an experiment for your students. Now we want to see the heartbeat of zebrafish before and after administration of the test drug. You can induce cardiotoxicity with the help of isoprenalin, and even you can measure the heartbeat also.
Video is not visible.
Okay, Mohammed. See this general anesthesia. So participant has to mute their mic. This is participant has to mute their mic. So this is various stages of general anesthesia. Stage of analgesia, stage of delirium.
First aim, tarvata
Participant. Now there is loss of. Sir, organizers, please mute the participant mic. Now you can say this general anesthesia activity.
Sorry.
Now the fish are transferred into a normal water. Okay. And it is transferred into normal water. Again, we will find the regaining its consciousness. This is full recovery. So this also you can demonstrate to your students. So I have given 3 experiments now. Anesthesia with cold water, anesthesia with 2% lichnocaine, then heartbeat of zebrafish, that is followed by the general anesthetics. Now, next experiment we are going is chemically induced epilepsy in zebrafish. So we can induce convulsions by putting in them either in picrotoxin solution or strychnine solution or nicotinamide solution. Any CNS stimulant on higher concentration produces convulsion. So the chemically induced seizure model in zebrafish, you will find many number of papers have been published in PubMed Index journal. And the activity related with the central nervous system like epilepsy, Alzheimer's disease or Parkinson's disease, where we can find this comparative chart. The seizure induced by pentylene tetrazole in the adult zebrafish, a detailed behavioral characterization. With this score will be there, when it is short swim, 0, increased swimming activity, 1, erratic movement is 2, circular movement is 3, clonic seizure-like behavior is 4. When animals comes into the fall to the bottom of the tank, that is the score 5, and death is score 6. So this is after putting the fish in the what you call the convulsion containing solution. Now, this is again the same thing. I will be explaining the convulsions produced by picrotoxin in our laboratory. Whatever we have done in our this I am showing you. So this is erratic movement of the fish. Now you will get the convulsion, right? The circling movement, erratic movement, and also the convulsions. Again, I am playing the same video. Just you have to listen the video. Erratic movement, circular movement, and convulsions after picrotoxin. So this is erratic movement. Now it is convulsion. then you will find circular movement. So for each and everything, so we have a score, as I have explained previously. Okay, so now if you have protoxin, strychnine, nicothamide, even in puffin in your lab, the pair of fish cost only 15 rupees. Only 15 rupees. We can get the fish. Then you can explain to your students. Right. Now, Zebrafish model to study diabetes. How the diabetes can be induced. Diabetes can be easily induced in animals, in fish. Whereas in rat and mice, you have to go for streptozotocin or the aloxone. If you give these chemicals, what will happen? There will be mortality. Means many animals will die because of the high glucose concentration. So to avoid this, the zebrafish can be used for studying anti-diabetic activity. I have given some similarity here. Alpha, beta, delta cells, same in case of fish as well as the human. Now, how will you find out that the animal is having diabetes or not? Okay. So for that, now we are going to take, after immersion in glucose water, for some times, you have to find out whether the diabetes is induced or not by using this experiment. Now we are going to remove the blood sample from the zebrafish. I said cold anesthesia. After cold anesthesia, to transfer the fish into this petri dish, you find out number of number of rays are there, from this top, the third layer. See now how my student is struggling to take blood sample from the zebrafish because it is very slippery, and if you see with the help of fine needle, on the third part, we require only 1 drop of blood. See, this is 1 drop of blood is enough to find out the blood glucose level with the glucose stream. Now the question is whether the fish alive or not. Definitely the fish will alive. Now we can see after taking blood sample, the fish will be transferred into the normal water. This is the normal water. And now the fish will regain its swimming characters. So this is normal water. After taking the blood sample, it is placed in normal water. Now slowly it regains. So this video will go some time to find out its what we call the regaining its position. Still it is the same condition. Now slowly, now it is 2 minutes 26 seconds the video. So automatically now you can see now the fish is alive. So don't worry, we can remove the blood sample, 1 drop of blood sample. Even daily you can take 1 drop of blood sample. Only thing is the skill is essential. So last week we have conducted an hands-on training in Vijaya College of Pharmacy in Dharmapuri, and hands-on training. So if anybody is interested, we can conduct the hands-on training also by using this particular technology. Now I just want to compare, only one comparison I would like to give. They can induce diabetes mellitus by simple immersion in high glucose water. High glucose water. So what do you mean by high glucose water? Then we have to go through the procedure. Procedure is with me. So if you place it in high glucose water, automatically there will be development of diabetes mellitus in a short interval. Even if you want to induce obesity, that is also possible. Sir, Nakupura sir, meeting like sir, please. All right. So now it is inexpensive. Already I told you what is the advantages of this. So these are the method by which the diabetes can be induced even in the fish. For example, you see incubation in glucose solution or overfeeding of zebrafish with the commercial food, alloxan exposure through incubation, intraperitoneal injection of streptozotocin, physical removal of pancreas. So now, why you want to take more risk? Just you put it in the glucose water, glucose immersion, there will be induction of the diabetes mellitus. Now, if you want to induce obesity, then high fat diet. We have to place the zebrafish in high fat diet. Then we can study what all things we are going to study. You will find here, you see, atherosclerosis, the blood vessel where we are going to remove the blood. I told you in the third line, from top, 1, 2, 3. 1, 2, 3. These two lines are up to end, and this line is very short. Okay, this line is also very short. See, sir, this I am explaining for the pharmacology people or anybody who is interested to work with zebrafish. When it is immersed in glucose water, there is chance of diabetic retinopathy, diabetic neuropathy, diabetic nephropathy, etc. When it is kept in high-fat diet, there will be development of atherosclerosis. You can see histopathologically, we can study the atherosclerosis. Suppose this is for wound healing. In case of diabetes, there will be impairment of the wound healing. That you know. Now, just you cut the fin, the size of the fin, just you cut it with the help of scissor. Okay. Then we can see your drug's effort to heal the wound. Suppose if you have any problem in removing the blood sample by this vein puncture, just you cut it here. Please see my cursor. You cut it here. That means down the fin. You cut it here. Fish, if you want, the fish has to be sacrificed. Right? If you want to take blood sample again and again, you go for puncturing the blood vessel. If you want to sacrifice your fish, then you can cut here so that this is the edge of the blood vessel. You will get the blood sample easily. Now we come to the central nervous system. The studies has to be studied in central nervous system. Just I will give the comparative statement. This is midbrain, hindbrain, and forebrain of human, and here it is the brain of the zebrafish, forebrain, midbrain, and hindbrain. So why I want to say is almost a similar parts we have. See, blood-brain barrier, the structure of blood-brain barrier in zebrafish, in rat, and in human. The drugs which cross blood-brain barrier in human will also cross blood-brain barrier of the rat as well as zebrafish because tight junctions are there, pericyte is there, in all basement membrane is are there, in all. The only thing is here neutrophil with astrocytic in case of human and rat, whereas here it is the radial glial process in case of the zebrafish. So, to study the drugs which cross blood-brain barrier, like anti-Parkinsonism, amnesia, psychotic or behavioral disturbances, everything is possible. But see, ALS, amyotrophic lateral sclerosis, Parkinson disease, Alzheimer's disease, Huntington disease, motor neuron disease can be studied by using the zebrafish. This is the way by which the brain injury is induced, but it is very difficult. I don't want to go for this. Now I will be explaining. Now time is 11:55. I will be taking 10 more minutes. The behavioral studies in zebrafish, how it will be progressed like that. We have the AnyMaze. This is a software, AnyMaze software. This has to be downloaded by proper registration. After registration, 2-3 days will take for the downloading. The software is called AnyMaze. So this is the trial software for 1 month, something like that. They will give. Again, you have to pay for this. So the advantages of this software is tracking the movement of the fish inside the maze. For example, here I will be explaining five experiments which I have designed in my lab itself. All these things are I designed in my lab. That's what I said to you. If you have this all, you can go for the hands-on training. Short-term memory, long-term memory, or anxiety, for example. So this is what you call the top and bottom. This is called novel diving tank. This novel diving tank, fourth one. 30 centimeter height, 30 centimeter length, and 15 centimeter width. So we have, this is the simple what you call it, the fish tank. So these have 50 liter capacity, and the water is filled with 20 centimeter. And this is divided by a line. This is bottom zone. This is the top zone. Okay. Now, if you place a fish in the tank, the preference of the fish always will be the bottom zone. It will not come to the top zone because of his anxiety. Now, we want to study the locomotor activity where distance traveled in meter, average velocity by in which speed it is moved, average velocity meter per seconds. Then anxiety-like behavior, latency in upper half, number of transition in upper half, and time spent in upper half. Now, you have to listen carefully. Now I am playing this video, the experiment which I have carried out in our laboratory. Now, see here one, this is the movement of the animal. The orange is movement of the animal. So the fish is there only in the lower bottom, not in the upper side. So keep the animals for 2 minutes or as per just to see the procedure. Okay. So almost the animals spend more time in bottom. So now, see, only by visualization is it possible? No. This video can be uploaded in the AnyMaze. I said AnyMaze is the software. Just I don't want to spend much more time to see this. I'm going to the next slide. You will find this is the movement of the fish inside the tank. This is the lower half and this is the upper half. Whereas here we will find animals spend more time only in the lower half, not in the upper half. After administration of the anti-anxiety drugs, you will find the animals spend time both in lower half as well as the upper half. So here we have number of readings, number of entries in bottom as well as upper, time spent by the animal, and what is the average velocity. So everything is possible when this video is uploaded in the AnyMaze, synchronized. Even how to take video, I will explain how to take video. Right. If any doubt, you can interfere, no problem. Now this is light dark compartment test. Same tank. This is tank measurement. I have given earlier slides. I have given the measurement. I have divided into two half with the help of a dark chart. I have made it into a dark compartment. This is dark compartment and this is a light compartment. This is light dark exploration model. Now, if you place the animals in this white compartment, that is light zone, automatically it will goes into the dark compartment because animals will prefer only the dark compartment due to its anxiety. When the animals are treated with anti-anxiety like drugs, benzodiazepine, then preference of the animals will be more even in the light arm. See, now we are going to study number of entries in dark compartment and light compartment, time spent in dark compartment and time spent in light compartment. It is recorded for 5 minutes, and this is the recording. So this is the light dark experiment. This is the camera stand. Here I have kept everything done by my student, PhD student. This is the camera. So the camera will, so because no need to disturb the animal, no need to stand nearby the fish. We can set it, and it can come out for 5 minutes. After 5 minutes, put on the video and upload it in the animes. So that shows like this. Here you can see I am putting the animal in the light compartment. Okay. Now the orange color is the fish. Now after a certain period, it goes into the dark compartment and then it will be remaining only in the dark compartment. Now it is entered into the dark compartment. Now you can see when it enters into the dark compartment, the average velocity is more. The speed of the animal is more. So number of entries in light compartment as well as dark compartment we can record. Okay, then I will be skipping all these things. Now you can see this is the movement of the animal inside the light compartment and dark compartment. And this is possible by means of enemies. Okay, and this is light compartment and this is the dark compartment. The result will get how much time spent by the fish, the light compartment and dark compartment. There is no bias. Everything is, there's no fabrication of the result. You have video and video is uploaded in the animes, so you will get proper. Now, novel object recognition test. This is to find out the memory capacity by training. Okay. Now, this is the habituation phase. Zebrafish explored for 5 minutes 2 times a day. Then, duration of the experiment is 4 days. Habituation, training, and testing. Now, habituation is over. Now, I am coming for training phase. Where I have divided the light and dark compartment on the same tank I am using here. I am keeping 2 objects. This is one object and this is second object. Both are identical. So allow the zebrafish to explore for 10 minutes with the 2 identical objects. Okay, after the training phase. Now I want to go for testing phase by administration of the test drug. Whether animal is having memory retaining capacity or memory deficit capacity. After 1 hour, one familiar object. This is green trees or familiar object. So this here, I have removed this green tree with one waterman. So now object is changed. This is called novel object. Whether the animal can able to recognize the novel object. If it recognize the novel object, then it have memory deficit activity. For example, memory enhancement, memory deficit, both can be studied by using this method. For example, here we can see, I am playing this video. See, 2 novel objects are there. Okay, 2 novel objects are there. I am putting the fish. Putting the fish inside. Now the movement of the fish will be synchronized. You can see how the movement of the fish takes place in and around the object. So it will be only in the object. I think the participant can able to understand because almost all are staff members. Okay, better understanding, I can go to the next slide. See here, we see during the training phase, this is green tree, this is green tree training phase. So animals will be there in and around this object. Whereas in testing phase, I have changed this object with the novel object, waterman. So this time, the animal is unable to swim, or it is not at all nearing the object because it is a novel object. Is unable to recognize the novel object because of the training. It unable to recognize the novel object. Suppose if we give the what we call the scopolamine produces memory deficit. So automatically the animal's movement will be there both in normal object as well as the novel object. The concept, I think, student staff can able to understand the concept. Now the last experiment I am going to explain is inhibitory avoidance test. This is my PhD student. He is a very good, sincere student of mine, and he is doing work with anti-Alzheimer activity. The same light and dark exploration model. So what happens when the animals comes into the dark zone? No, we will be applying the shock with the help of electroconvulsion meter. Electroconvulsion meter will be applying the shock. What I said, usually animal prefer only the dark compartment. So whenever the animal comes into the dark compartment, if we give an electrical shock, then thereafter animal will not comes into the what you call dark zone. This is called inhibitory avoidance test. So I will show you this also. I said you can see this 1.4 minutes video. So 1.4 minutes video you have to see, putting the animals in the light compartment. See here I have bifurcated with the help of a barrier here. The same apparatus, we have multiple utility. Okay, 1 minute attest time is with the light. Then slowly you have to remove the barrier. If you remove the barrier, automatically it goes into the dark. Okay, after 1 minute. So now it is in the dark. So we are going to apply the electrical shock in the TV. Now you just remove the barrier. After 1 minute, barrier is removed. So the fish will goes into the dark compartment. Now it is entered into the dark compartment. Okay. Now in the dark compartment, we are going to give electrical shock. Now, the help of the electroconvulsion meter. If we give electrical shock, the movement of the animal we can see here. See, this is the shock after application of the shock. So now what happens? The animal learns that if we enter into the dark compartment, the shock will come. Okay, this is called inhibitory avoidance. So if you give antipsychotic drugs, chlorpromazine, it forget whatever it learns so far. So this is antipsychotic or anti-amnesia activity, etc. Now, this is the last experiment. Within 2-3 minutes, I will be completing. This is called T-maze. All right. Everything made up of glass only. Water will be there. This I made it into dark, and this I made it into green. This is red and this is green. In sense, this is unfavorable zone. This is the favorable zone. Now I am placing the animal in the starting zone. Okay. Now the movement of the animal will be there. Whenever animal comes into the unfavorable zone, and we will be shaking it with the help of glass rod. Just to stir it with the help of glass rod. So some kind of stress or some kind of disturbances will be there when animals enter into the red zone, danger zone. Whereas if it goes into green, and there will be reward. some kind of food we will be giving. So this is our habituation phase, right? Now we are putting the animal in this zone. This is the animal's zone. After acclimatization, this barrier has to be removed. So animal will move here and there. One dark zone is there and one light zone is there. Okay. Now we'll see time spent by the animals in this is the danger zone. This is the reward zone. This is the danger zone. So the fish will prefer only the reward zone, not the danger zone. After administration of the antipsychotic drugs or anti-amnesic drugs, the movement of the animal uniformly will be there both in both in danger zone as well as the favorable zone. So with this, I am concluding my lecture. If you have any doubt, you can ask, please. With this, I am concluding my lecture. If we have any queries, we can ask, please. Still, some more experiments are there, but due to lack of time, I am concluding my lecture.
Sir, thank you very much for your wonderful presentation.
Thank you, sir. Thank you.
You have given us a very good insight into the zebrafish model as a research alternative for the rodent research in the pharmaceutical and pharmacological experiments. And one of the participants has asked, sir, is cold water sufficient for inducing anesthesia?
Yes, yes. Cold water anesthesia is one of the procedure, right? Only thing is just to make the ice cubes before starting the experiment. Ice cubes has to be converted into the water liquid. Okay, not the cold water in sense, not from the refrigerator.
Okay.
Just to make the ice and convert ice into water, just to prior to the experiment, then you can put it.
Sir, can we use any, the same participant, they asked, can we use any anesthesia agents and all?
Yes, the one list of table you have anesthesia, euthanasia, analgesia used for zebrafish, but due to lack of time, I excluded those slides. Certain procedures are there.
Sir, one more participant asked, sir, is there any specialized equipment necessary to handle, sir, to perform surgeries on these zebrafish?
Sir, actually, some instruments are available commercially, but so I am working in a government institution, so I have made it by my own. So all the experiments are, all the instruments were done, fabricated by me only. Then, sir, why fish moves equally into two zones? Then, I think question asked by one of the participants. So after administration of the anti-amnesic drugs, if there is loss of memory, then the fish will move both the zone. When the memory is retained, then animal will goes only to the favorable zone, not unfavorable zone. So our aim is to find out whether amnesia is induced in animals or not, and our drugs is having anti-amnesic activity or not. Any message? I think message I can from Navya Manyala said, thank you, sir. I think I am not able to see the what do you call it, the messages. If I see the message, I can give you the answer.
Yes, sir. Participants, you are free to ask the questions.
Okay. Any questions?
Sir,
82 participants are there. Only 4 questions I have
my
faced.
question, sir.
Please.
Sir, you said about hands-on training workshop.
Yes.
So we need to come over this PGIMR and
Only this video, and also if the instruments are there, after any means download, you can go for hands-on training.
Okay, sir.
How
Sir,
to remove the blood sample, how to remove the brain from the fish for neurotransmitter studies.
yes, sir. Sir, you have
Okay,
showed Animase software, na, sir.
Anime software, yes.
So is it freely available or we need to purchase that Animase?
That's what I told you. No, we have to register it. After registration, 2-3 days will come, and this is a trial version for 1 month.
Okay, sir. Sir, one more question from one of our faculty member, Dr. Ramayappa.
Yes.
Sir, he's asking, is ethical committee permission required for handling?
Yes, sir. Yes, but it is very easy to get the ethical clearance. Unlike rat and mice, even without ethical committee clearance, also we can go for studies if it is PG or UG demonstration. Suppose if it is PhD work, then unnecessarily the examiner will ask where is the clearance certificate. Even if you apply for the clearance, it will be given instantly because there is no invasive or no one will worry about the utility of number of fish because one fertilization it gives 300 larvae.
Sir, another question from the Dr. Ramayappa. So, is there any, from where we can get this zebrafish?
Sir, anywhere you can get, even not only zebrafish, even ornamental fish can also be used.
Even ornamental fish is also.
Yes. If you go to these hatchery farms or fish who provide the fish, then you can get it.
Sir, this is my question because when I'm in that pharmacological research, they told me to select only albino Vista rats and all.
Mm.
Like that, is there any specific breed in this zebrafish or?
Okay, I will come to Navya Manyala asked, sir, can we do PhD research by using Zebrafish? If it's so, in which university we can do? Sir, there is no question of university. It depends upon your guide. With Zebrafish itself, we can complete your project work. No, don't worry. With the Zebrafish itself, we can complete your studies if you go extensively. And sir, what is your question, please? Okay. Albino, right? Why Vista rat? Okay, so if you have, either you can do it zebrafish or rat. Okay, if you are comfortable with your monetary fund, if you have, so I said, one rat will cost 360 rupees, whereas one fish costs only 7 rupees. So if you have enough money, and you can do experiment with your rat also. Handling of rat, so many things are there. Yes.
I think another participant is trying to ask a question. Present market. Still any questions?
Swapna, Ramadevi, Sheikh Hasina Baham, Divya, Manogna, Anirudh, Sunita. If you have any questions, you can ask. I think Navya Manyal asked some questions. Kalyani, Muhammad Usman, Deepthi, Nagasam, Vishal, Jawahar Babu. Any questions? Okay, thank you very much for the opportunity. Thank you.
Sir, thank you very much for presenting an excellent research topic for our participants. And I thank you from the bottom of my heart from on behalf of Sri Vishnu College of Pharmacy and all our staff members. Thank you very much, sir.
Okay, my mobile number is 9443085956. 9443085956. And my mail ID is DRS Kavimani, Dr. S. Kavimani, DRS Kavimani @ yahoo.co.in.
Sure, sir.
Yahoo.co.in. If
Sharing with
any,
all participants. And
Any help regarding this, any doubt about the zebrafish, or even if you want to give me lecture, I am ready. Okay, thank you very much.
Thank you very much, sir. So with this one,
Sir, one can I ask my phone number and mail ID? Please forward my mail ID and the phone number to all participants. Okay, sir.
Okay, thank you, sir.
Thank you. Okay, bye. Thank you. I am leaving. Thank you.
Dear participants, with this one, today we are concluding the session. So please join us at the same time tomorrow for the tomorrow session. Thank you, sir.
Thank you, Pratisha, for acknowledgement. From Acer to I have acknowledged. Thank you. I am leaving. Thank you.